Good afternoon. Originally, I'm the last speaker. Now I'm moving to the before like that minister talk. So I would like to thank Jeff and the foundation, also the university colleagues to inviting me here. I got very impressed. This is my first time to get this country, but I find the people here very friendly. Everything is good. Okay, we're moving to another, when we talk about the environmental related, we move to another topic, that's the foodborne toxicants. So specifically, we focus on the natural occurring foodborne toxins, specifically is aflatoxin. I will introduce some background, then mainly focus on in past 25 years, our study in the many countries globally. So focus on the aflatoxin exposure in mothers and children. A lot of studies linked to the with the pregnant mom and the 2 years older children. So mycotoxins are the most important category of health-related foodborne toxins. They are toxic agents mainly produced by fungi genera for the three major genera, Aspergillus, Physarum, and Penicillium. Currently has been like 400 different kind of toxicants been identified and reported. Majority those are produced by laboratory. So they inspired by the discovery of aflatoxin in 1960s because of there's 100,000 turkey died in the south of England. So then later on find mainly responsible parts, it's the toxin produced in Brazil and Uganda. They exported to the England as the feed components. They find that. So after that, there's a big effort to identify the different kind of these toxins. So aflatoxins, trichocecains, ochratoxins, fulminescents, and zearalenone. They find that involved in various human and animal disease. As shown in this picture, all those like different growth appearance, Physarum, Penicillium, Aspergillus. They are produce spore on the microscope. You can see that as a beautiful color there. So mycotoxin, they got the emphasized for research and management, not only because of their toxicity, but because of their economically impact. According to the Food and Agriculture Organization, that's in United Nations, the 25% of the world food crops are affected by mycotoxin every year. Global loss of food stuff due to mycotoxins is ranged of about 1,000 metric tons each year. So about 100 billion of exported commodities all over the world are susceptible to mycotoxin contamination. In the world, about over 120 countries and regions developed regulations to control mycotoxins. So in the US and Canada, they said at least they consider least contamination with aflatoxin, but still there's a $5 billion loss annually for that. So aflatoxin specifically, it's the representative of this mycotoxin category. They produced, as I said, Aspergillus flavus and Aspergillus parasiticus. The aflatoxin A, standard Aspergillus, standard flavus toxin. That the name comes out for that. Under UV lights, the aflatoxin, some aflatoxin show blue, they give the name called aflatoxin B. They show green, called aflatoxin G. Then they find in the aflatoxin contaminated feed, if they used for cow or sheep or those other in the milk, they have another derivatives, they call it aflatoxin M. So that's the name comes from there. So they caused a serious public health and food safety concerns, mainly in developing nations. Nowadays, the developed countries, even European communities had some problems almost every year. Specifically, the European Union, they expanding, including the former Yugoslavia, those countries, they had issues every year. There's a newspapers as a medium covered that. So more and more, there's a lot of animal aflatoxicosis, like turkey X disease, that's originally called that. Actually, now more affect almost every animals, chicken, duck, goats, all can infected by those aflatoxins. More importantly, aflatoxin linked to the chronic human disease like liver cancer. That's since 1963, find the aflatoxin can induce the animal cancer. After that, there's a tremendous studies, including our group, for now clearly established aflatoxin is the group 1 human carcinogen for that. So because there's not too much knowledge about a human acute aflatoxis until 2004, 2005, there's about 350 people got acute aflatoxicosis. There's 220 people died on that in East Kenya. So then people realized, hey, acute aflatoxicosis, it's the big issue. Later on, even until today, there's a report in Tanzania or other those countries, they have kids died of aflatoxicosis for that. So when I become independent, I'm more focused on how aflatoxin affect the human immune system. So we did a lot of studies, but it's not a topic for this talk. Further, we find that the aflatoxin exposure can anti-nutritional effects. So they can aggregate the malnutrition issues in children. So these are those natural occurring aflatoxins, B1, B2, G1, G2, M1, M2, and those two major ones are listed here for you guys. Moreover, this is the traditional in 70s, they find in the north and south 30-degree altitude, the Aspergillus flavus, the toxigenic fungi grow. So you see a lot of countries out of that, they say, oh, that's not our problems. Unfortunately, the global warming climate changes, they extended that. Now, almost, you see, 85% of the region of the globe, they detected the toxigenic aflatoxin, Aspergillus. So based on IAC, the International Agency for Research on Cancer, they summarize or average all over the world study. They find different countries from their maize products have contaminated with aflatoxin. Like this is in the maize products, this is in the food. They measured like a human actually intake, that's levels there. So this picture is Jerry Wogan. It's a longtime MIT faculty and serving as MIT department chair for 20 years. He's the pioneer of aflatoxin research. When he heard the news in the South London, 100,000 turkey died, he immediately booked the air tickets, fly there, collect the samples back to Boston. While he isolated those fungi in the lab, established protocols, he persuaded a young faculty called George Bucke, and from, actually it's immigrants from Czechoslovakia. He agreed to work with him. Eventually, as the prominent chemist, they synthesized all those aflatoxins. Make a Wogan's lab is the only lab in the world. They have materials to conduct all this kind of researches. So their studies, almost a Wogan, there's like 30 PhD students working out all the metabolic pathways, the carcinogenic dose response or effect, everything with that. Left all the human studies, we use those to continue to work on that. So nowadays, we know every metabolic pathway. So my lab use this kind of pathway, can use that as a model to test new chemicals, what the toxicity for that. We use the biomarkers like aflatoxin M1, in the urine or milk, they can serving as a biomarker. Or those aflatoxin binding to the lysine and albumin, we can use the blood that level to represent the long-term exposure for that. So those are really biomarker used for that. Since there's a lot of students here, what's the current of research focus? I just share with you guys. Well, a lot of folks in the last 15 years put how they related to the children's growth and development, especially stunting in the Western. Then the immune toxic effect, the linkage to the human chronic infections, also acute infections. I collaborate with Kenya, like the pediatric department and the universities. We find aflatoxin exposed children, their blood antibody levels, they got a vaccine, all the kids get a vaccine, but high aflatoxin levels, their antibody levels lower compared with to those lower aflatoxin exposure. That's based on that, they submitted the Wellcome Foundation grant. We got 3 cycles review, but only last month they said, well, you need to show more evidence to see. We only show 2 antibodies. Now they said want us to do the preliminary studies for more. Then they will consider funding for us. Moreover, we know that there's a lot of interactions between aflatoxin, like another toxin called fumonisin, even ubiquitous in the corn products. They can synergistically affect on humans. On the other hand, there's a 2 focus on prevention. biological control. There's a woman in Kenya, I guess, got a World Food Prize because he found that in Kenya soil isolated the competitive strains with aspergillus. They got the World Food Prize. That's called a biological control. Then chemical control, a lot of people use the binders, like they can reduce the exposure on that. In our lab, mainly use the biomarker-based, like USAID, United States Agency for Research on Cancer, supported my lab in the last 25 years. They wanted me to establish a global network for aflatoxin exposure in children and in the pregnant moms for that. So they try to link it to aflatoxin exposure with nutritional deficiency, growth inhibition, and infectious disease. They also want to use the biomarker to investigate it, to test the efficacy of different intervention strategies. That's the study for that. So in my lab, for many years, we're based on those analytical instruments available, HPLC, fluorescence-based. Recently, we last several linked to the mass spectrum to can detect those different kind of aflatoxin biomarkers in human biospecimen for that. Here, I just want to briefly introduce our studies like in African countries. So you can see in the top, we work with Burkina Faso, then Ghana, Kenya, Malawi. Those mainly you can see the mother, usually pregnant mother, children, the infants, the newborn, children generally is lower than 3 years. So those levels, you can see the detection rates here. From 79 to 100, they're available. Look in the median, there's a big variations on that. Then the other countries, like Malawi, pregnant women, Tanzania, they didn't define that that's like it's the children or those. But Zimbabwe, they have pregnant women and the children. So that's the. Then we collaborated with Uganda. Why Uganda? Because in 1980s, Uganda was very prevalent for HIV. So they asked the world help. The British Medical Council established a cohort study involving 24 villages, about 20,000 people. US, NIH, and Johns Hopkins established their own cohort study there. Then other universities established another cohort. All the cohort study there. If you want to evaluate aflatoxin with infectious disease, you have to go to Uganda. So unfortunately, first I contacted Uganda government. They said, no, we are not interested in that. Then I said, forget it. After 2 years, they sent me a letter, invited me to visit there. They said they really want to work with me. So they gave the examples. You see how many lungs there. There's 1, 2, 3, 4, 5, 6, 7. Each different amounts collected from their cohort study. My lab analyzed those aflatoxins for that. You can see that's the summary of that cohort, the study. Their medium, it's not very high compared with Burkina Faso and those others, but it's still detectable for that. That's the Uganda. Then the international research on food, International Institute for Food Research, they contacted me and said that they got funds from Gates Foundation and Wellcome Foundation to conduct the study, initiate a cohort in pregnant women and those infants. So we signed a contract with, that's in East Kenya. East Kenya is the most contaminated area. You guys can see that at the medium. It's very, very high. More, okay, I do. You see, those higher over those 40, they reach the aflatoxicosis level. So we give them a warning for that. So this is the same, you see, can see the levels for that, very, very high there. So we further, you can see those levels, clearly there's a variation for that. So in addition to the Africa countries, we did a study in Asia and the North, like in the South America. For that, you guys can see, even US, China, Haiti, Malaysia, Mexico, Pakistan, we got all detecting those collaborating. That's in Mexico, you see the newborns. They are different, it's in 4 months and 12 months data. They even have different area in Mexico. That's in the East Timor-Leste we conducted with, that's funded by UNICEF for us to evaluating their mother and the children exposure for that. More and more, there's a Harvard and Nepal, they have a cohort study called aflatoxin birth cohort study. We use that to detect the aflatoxin B1 lysine F. There's a multiple points. I only list the first points there. Then we also, they collected the breast milk from mothers. We measured the aflatoxin M1, the detection levels, it's 93. You can see they also have the examples in different, the dry blood spots. We try to, give me 1 more minute. So that's in the Nepal cohort study. Well, although it's past time, I want to still take the clearly the covariant adjusted regression model, they find that the aflatoxin exposure in infants significantly associated with lower Z-score. Similarly, they also, with those the weight, those Z-scores for that. So time lag, the aflatoxin B1 concentration is associated with the growth inhibition for that. I guess that we not only did the aflatoxins, we also, in that Nepal cohort, we detected other mycotoxins for that, like ochratoxin, fumonisin, and the deoxynivalenol for that. So that's all those results here. So I would thank the worldwide collaborators in last 25 years with me. Also major funding from USAID, also NIH, several grants also for that. So I feel very happy to collaborate with Ecuador project. They are funded by the foundation. So thank you very much. I would like to take some questions.